All right, so this is Chapter 2, The Chemistry of Life. So the dog, water, air, all forms of matter, everything is made up of chemicals. Everything is made up of atoms. So matter is any material that takes up space. Generally, you're going to have one or more elements in anything. So like if we looked even at like the water, it'd be the most simplified version or simplified example from what we have up here. The water is made up of hydrogen and oxygen. So an element is a substance that can't be broken down any further by any chemical means into other substances. So yes, they contain subatomic particles, but they can't be made into another substance. And then here's the periodic table, and I just want to kind of point out a couple of things. So you have your atomic number, the element name, the symbol, the atomic weight. So depending on the periodic table, you'll have, more or less information. The basics are the atomic number. You might just have the symbol. So the chemical symbol is unique for each element. If you look at number 1, hydrogen, and number 2, helium over here, they both start with H's, so they both couldn't be H. So hydrogen got H, whereas helium got HE. And then some of them get crazy. If we look down here, like at potassium, number 19, potassium is abbreviated with K, or like iron is Fe. Some of these go into like the Latin names of the elements. So I already mentioned all this, taking the full name, the symbol, and then the atomic number and the atomic weight. We're gonna, before we get more into that, we're gonna talk about protons, neutrons, and electrons. So in an atom, you have a nucleus, and then you have the electrons orbiting around that. So an atom is the smallest piece of an element that has the characteristics of that element. So the 3 smaller particles I already mentioned: protons, neutrons, electrons, also sometimes called, or you might hear, subatomic particles, sub meaning below, so lower than the atom level, subatomic. So our protons and neutrons are going to be in the nucleus, at the very center here. And the electrons are going to be orbiting around very fast, and they're very, very small. So here's a good table to kind of show you. So an electron has a negative charge. We're going to say the mass is 0. It does have mass, but it is so small that it's insignificant here. And it's surrounding the nucleus, whereas the neutron and the proton are in the nucleus. The neutron and the proton both have a mass of 1. So when we calculate the atomic mass, it's the number of neutrons plus the number of protons. Neutrons are neutral, there's no charge on them, and protons are positive. When the number of protons equals the number of electrons, then the charges cancel each other out. If you have more electrons than protons, then the overall charge of the atom is negative. And if you start missing electrons, protons aren't going to go anywhere. But if you start missing electrons and you have more protons than electrons, then it's positive. We'll get into that a little bit more. So when we look at back at the periodic table, if we look at the atomic number, that tells us how many protons are in each atom of that element. The proton number is unique to each element. So in carbon, there are 6 protons because its atomic number is 6. So the mass is the total number of the protons and neutrons. I already said that because they each have an atomic mass of 1. So if we add proton and neutron, we get 12 for this one. So there are 12, or the mass number, I should say, is 12. So we also need to take into account the isotope. So when we go back to the periodic table, you'll see that a lot of them have decimal places. Well, how can it have like 0.09 mass if a proton is 1 and a neutron is 1? And this is where it comes into play. So an isotope is different forms of the element. If you look at this, you see that the protons are all the same. So you have carbon 12, carbon 13, carbon 14, and they each have 6 protons. They each have 6 electrons because they're neutral atoms. So the number of positives and the number of negatives cancel each other out. What changes is the number of neutrons. So in carbon 12, which is 99% of all carbon atoms, there are 6 protons, 6 neutrons, 6 electrons. Carbon 13, it's carbon 13 because 6 plus 7 is 13. So this time you have 7 neutrons. You have an extra neutron in there. And then carbon 14, 6 plus 8 is 14. That's where carbon 14 comes in. It is 8 neutrons and 8 protons. So when we take that number, the varying mass numbers of the isotopes, 12, 13, and 14, we're going to average them out, and that's how we get the number we see on here, the atomic weight we see on here. We take the average mass of all the isotopes and then get the atomic weight. Now, when you're calculating things like the number of protons, the number of neutrons, well, the number of protons you'd already know because of the atomic number. But if I said, hey, tell me the number of neutrons in carbon, just take the whole value. Don't worry about 12.012. Take 12 minus 6, and then you get 6 neutrons. So most carbon isotopes have a mass of 12. Yes, there's 13 and 14 for carbon 13, carbon 14, but we're going to take the average for that atomic weight. All right, so for this one, the atomic mass of nitrogen is near 14. We're just going to say it's 14. And then indicating that most nitrogen atoms have a mass number of 14, how many neutrons does the average nitrogen atom have? So you can pause and kind of work this out on your own, but remember, and I know you can't really see the whole thing because of the way they put the video for me here, but you know, the number you're missing in here is 14 and some decimal places, but we need to just use 14. So we know it has an atomic mass of 14. We also know that its atomic number is 7. And remember the atomic mass, which I'm just going to abbreviate AM. AM is equal to the number of protons plus the number of neutrons. This is our number of protons right here. So if we know this is 7 and we know this is 14, then we can calculate the number of neutrons. So 14 minus 7 gives us 7. And then I'll go to the next slide and you can see that that is the correct answer. All right. Chemical bonds link atoms. So like in this water molecule, for this oxygen to be connected to this hydrogen, there has to be a bond here. Same thing between this oxygen and this other hydrogen, or the oxygen and the other hydrogen. So to hold them together, we have chemical bonds. And electrons, the things orbiting the nucleus, are what determine bonding. If there are empty spaces, the atom isn't very stable. It wants to have a full outer shell, and depending on what level of shell it has depends on how many openings it has. So like hydrogen only has one shell because it has one electron right here, but it has one vacancy, so we can bond with one other thing. Whereas carbon actually has the room to bond with 4 things, or I should say up to 4 atoms. That was a really bad circle. Nitrogen only has 3 openings, and I think that's oxygen. I can't remember what was my last one. I think it's oxygen, and there's like 2. Yeah, I think so. So let's talk about, so this outer shell is called a valence shell. That's that last shell. So that's the one that matters for bonding. So like even though this carbon has 2 shells, this one doesn't matter because it's full. It doesn't matter if it had 5 shells. It's the last one that is bonding with other atoms. So the unpaired electrons form, atoms are most stable. I've already said that when there's no vacancies, bonding with other atoms fills those vacancies. So we have a carbon atom and 4 hydrogen atoms. Remember, each hydrogen atom has space for 1 bonding. So the carbon atom has space for 4. We have 1, 2, 3, 4, which is why it bonds in this arrangement that you can't really see because of my video, but you can go back and pull up the PowerPoint, or pause and pull up the PowerPoint if you haven't already, so that way you can see it. But we did discuss this in class. So everything's even, everything's spread out. So the 4 hydrogen atoms share their electrons with 1 carbon atom. All 5 atoms fill that outer shell, so now they're all happy because they're full. Look at how their outer shells overlap to form this molecule. You see some overlap here, so that they can share their electrons. When atoms share electrons, it's called a covalent bond. Now, if they share them equally, such as the case with this methane molecule, then this is a non-polar covalent bond. Non-polar. And the reason why it's called non-polar, again, they are shared equally, and there's no charges on this. This is a neutral molecule. There's no charge on it. And you know we're going to look at one that's unequally shared and has a charge. So there can be, covalent bonds can actually do more than just 1 bond. So let's look at the oxygen atom here. So oxygen has 2 vacancies. So we have 2 atoms here. And what they do is they actually do a double covalent bond and share these 4 between each other. So this is O2. This is another way to write it. This double line here, right there, means it's a double bond, but they're sharing 2 electrons between those 2 atoms. Let's talk about water. So water is very similar to methane. You have, this time you have an oxygen atom and you have 2 hydrogen atoms. And what it's going to do, and I'm going to kind of sketch it over here, is between each hydrogen atom and the oxygen, they're sharing 2 electrons, and you can see it right here in this part of the picture. But oxygen pulls a little bit harder on those electrons, and you can actually see there's some electrons over here. So this side ends up being negative, and this side ends up being positive. And even though they're single covalent bonds, they are polar covalent bonds. Polar covalent bonds. I'm just going to write CV because I don't feel like taking the time to write out covalent. And that refers to the charges on the end. You have a positive and a negative here. And this is why water is so special, because it has those charges. It's able to dissolve a lot of substances because of that, and other things. We'll get to it. So again, in water, the oxygen pulls the electrons more strongly than the hydrogen atoms. And then this, we talk about electronegativity. So it's the ability of an atom to attract electrons. So let's get into that a little bit. So on this side, there's increasing electronegativity, but decreasing atomic radius. So this has a smaller radius. That's a really bad circle. And then these are, we're talking about larger, so there's a larger radius here. So increasing electronegativity and decreasing atomic radius. So we're talking about ones with larger radiuses versus smaller radiuses. It's, the gist of it is the number here is going to help you to figure it out. So carbon and hydrogen, here's the electronegativity number for hydrogen. Here's the electronegativity number for carbon. they're close to each other, 2.2 versus 2.6. So neither one is pulling more strongly on the electrons. They're pretty equal, which is why our methane molecule here is neutral. So these are those non-polar covalent bonds. There's no charge on the molecule. It's a neutral molecule. However, when we look at hydrogen and oxygen, now we have 3.4 for oxygen and 2.2 for hydrogen. So oxygen has a higher electronegativity number, so it can more strongly pull those electrons. And the result is that negative charge on the oxygen side and a little bit of a positive charge, a very bad plus sign, on the hydrogen side. So this is where we call water is a polar molecule. And we say these are polar covalent bonds because they result in charges on the poles of the atom. So think of like a battery. You have a positive and a negative. You go to put it in your remote control for your TV. You have to make sure they're the right way. So polar means there's a charge. We can say it has polarity. And these are held together by polar covalent bonds. So charge difference between the oxygen and the hydrogen sides of the molecule. All right, so if we were to look at the molecules in like a bottle of water, the molecules are moving, and what they are doing is they are actually, the positive end of, let's say this one, is being attracted to the negative end of this one, and this results in these hydrogen bonds right there. And they are weak attractions between positive and negative. And they form and they break and they form and they break. There's no electrons being shared here. It's just like magnets sticking together and being pulled apart. Same kind of idea. So again, the slight positive charge on one atom, on the hydrogen of one molecule, attracts the negative charge on another molecule, and they make these hydrogen bonds. And these become important because they keep water together, but they also create surface tension. And it can also allow water to stick to other things, and we'll get to that. So hydrogen bonds give water its unique properties. It's also going to be important in DNA structure and in proteins. All right, some atoms have such different electronegativities that one will just pull its electron from it. So let's look at sodium, Na, and chlorine, Cl. You have 0.9 and 3.2. They are so different that that chlorine ion or atom is going to pull when they join together. So sodium chloride is table salt. When they bond, when these two atoms bond, this electron is actually going to be given to this one, which now means there's going to be an extra proton here. So this is going to cause this to be positive. Now this one's going to have an extra negative. So this one's going to have more electrons than protons, which means this atom's going to be negative. The sodium's going to have more protons than electrons, so it's going to be positive. And when atoms donate or gain electrons, it's called an ionic bond, and it's kind of cut off here somewhere. Write it I-N. Whoops. What in the world? There we go. I cannot, oh, and I picked the wrong color. Ionic. There we go. Ionic bond. So again, they still are going to achieve that full outer shell. Since this one is getting rid of one, now it doesn't have a vacancy. That outer shell, it only had one to begin with. So now its outer shell is full, and now the chlorine is happy because its outer shell is full. Here's a great breakdown for it. I'm not going to read through the whole thing. Check it out in the PowerPoint. Pause the video if you need to. This breaks it down. It tells you how strong or weak they are. Hydrogen bonds are the weakest, but their strength in numbers. A little bit more about the electronegativity. So nitrogen has 3 vacancies in its outer electron shell. What type of bond might nitrogen form with hydrogen? How many hydrogen bonds would one nitrogen atom bind? You might need to reference the electronegativity scale, which was figure 2.6. So give you a second to read that and take your pick. And it's covalent bond with 3 hydrogen atoms because it has 3 vacancies. Look at the electronegativity scale, they're pretty close. I'm going to move on. So hydrogen bonds that hold water together help to give it properties. Realistically, it's the polarity that gives a lot of properties, ultimately. Cohesion is the tendency of water molecules to stick to one another. This is really through those hydrogen bonds. They're sticking through hydrogen bonds. So cohesion, right here, this is the hydrogen bonds. This is the cohesion. This allows a surface tension so that you can have some insects can actually walk on the water. And you see that there's an indent here. It's not in the water, it's on the water. I'm trying to find like some paper, so it's like actually you can see the indent because it's on it, not in it. So it's a little different than if you were floating like in a pool or in the ocean or something. You'd be like an iceberg, kind of in, kind of out. And this bug is actually sitting on top of it. Now, if these hydrogen bonds are disrupted, it could fall through, but it has evolved to be able to spread itself out and distribute its weight. Water is also adhesive. It looks like I struck that out. Nope. Let's see. Water is adhesive, meaning it sticks to other things. So we're talking about water going up a tree. Take the giant like redwoods in like California. They're very, very tall trees. They don't use any energy to get their water from their roots to their leaves. Water molecules go into the roots and they get pulled up the stem and out through the leaves. As water evaporates, it actually pulls the next one through. So when a water molecule evaporates, it's going to pull one up to its spot, and it's going to be a chain reaction and pull more and more and more. Now, if there's no water down here, then that's a problem for the plant. But the water sticks to the sides of the xylem in the plants. That's adhesion, and it sticks to each other. That's cohesion, and collectively we call this capillary action. So again, it allows it to climb to the, to the tree's roots to the highest leaves, and that was capillary action. Water is a good solvent. Sometimes you hear this called the universal solvent. So water dissolves hydro, this is water, philic, loving, solutes. If it dissolves in water, then it is either a polar solute or an ion. So like if we could dissolve salt in water, and we already talked about salt. Salt is NaCl, and it is a result of an ionic bond where this one is positive and this one is negative. Therefore, because they have charges in that molecule, water can react. You can have a water molecule, this is the positive and this is the negative. This side is going to attract this to pull it apart, and then this would attract that one and pull it apart. So if it dissolves in water, it's polar or a result of an ionic bond. There's some charges on the molecules. So again, the polarity of water helps molecules dissolve ions. Here's a better example of it where you can see the sodium here and the chloride here and the attraction between it. So slight negative charge water attracts the positive in, so we zoomed in a little bit more so you could see it. Notice how they're oriented around the sodium. The hydrogen are pointed away because they're the positive, and like repels like. Whereas on the chlorine ion over here, you see the hydrogens are oriented towards it because the negative charge of the chlorine atom attracts the positive side of the hydrogen atom, or hydrogen atoms, I should say. So water does not dissolve anything that's hydrophobic, so water fearing. If it does not dissolve in water, it is nonpolar. So think of like oil. Oil is nonpolar, which is why it will not dissolve in water. You can shake it and make a suspension and it'll kind of be suspended in there, but it doesn't actually dissolve into it. Because of the hydrogen bonds, water is able to resist sudden swings in temperature. So if we were to take the temperature of the air throughout the day, every hour on the hour, we would see it would change throughout the day. If we took the same measurement of a body of water, whether it's the Chesapeake Bay, whether it's a lake in your neighborhood, whatever, we're not going to see those fluctuations, especially if it's a larger, deeper thing of water. And this is why coastal regions tend to have milder temperatures. This is how sweating helps to cool us off, because those hydrogen bonds require energy to break them. So when you have sweat on you, the water is absorbing the heat from your body, and the more it absorbs, eventually it is able to evaporate, assuming there's not high humidity in the air. That's a totally different thing. And it will take the heat, remove the heat from you. Another thing is water expands when it freezes. Most things, when they become solid, they become more dense than their liquid form. Water's the opposite. So you see the liquid water down here in the arrangement of the molecules. They're pretty tightly packed together. They're able to slip and slide past each other. But as they slow down, when they become a solid, they actually form these nice arrangements, and they kind of push out away from each other, so that there's less in the same area. If we counted how many molecules were in each of these circles, there's going to be more on the liquid water than there is in the solid. And that's what we mean by less dense. There's less molecules in the same amount of space. And this is really good because it allows ice to float, which allows aquatic life to continue even when it's cold outside. It also does some insulation up here. So cold air going over this way is actually kind of kept from mixing down here, and it allows this to stay warmer than the ice up here. And it also, if it sunk, it would freeze, and then it would sink, it would freeze, and then it would sink, and eventually it would stack up. So even if it didn't get cold down here, if this sunk to the bottom, you would just have layer upon layer on top of it. Okay. And I apologize if I seem like I'm talking really fast. I'm trying not to make this a super long video, but I'm also trying to make sure I get everything in there. So water participates in various chemical reactions. Two big ones we're going to talk about: photosynthesis and cellular respiration. So it is an input for photosynthesis, and it is an output for cellular respiration, but very important for both in terms of being made or being used. All right, which property of water contributes to the high surface tension of water? Take a moment, think about it, pause the video if you need to. All right, I hope you paused it, thought about it, because the answer is all of the choices are correct. Hydrogen bonding between the water molecules is the same as the cohesion. So those are, in water, that's the same thing. And the covalent bonds cause the charges, which allow for the hydrogen bonding. So if these two are the same, then you have to pick those two. Then chances are it's E, all of them are correct. Ooh, all right, let's talk about pH. pH scale is based on the amount of hydrogen ions in a solution, H+. This is really a hydrogen atom that has lost its electron. It's really just a proton. So acidic solutions have low pH, but they have high H+ concentration. So there's more H+ here, and there's less H+ down here. Ooh, my S's are really bad. Yeah, you got the gist, I hope. So again, basic solutions have a higher pH but a low H+ concentration because they release OH negative ions. OH is hydroxide ions. Also understand that the pH scale measures that, the hydrogen ion concentration. Whether something is an acid or a base depends on what ions it releases, not where it is on the scale. The scale is really just to tell you which one's stronger than the others. So pH is important because if you go too far from it, you're going to die. I'm going to tell you right now, there's not much you can do to alter your pH, despite what certain water companies might have you believe, which is a good thing because your body needs to maintain homeostasis and keep that pH where it's at. If it fluctuates too much, your enzymes aren't going to work, and if your enzymes don't work, you're dead. There's no way around it. So this is where, buffer solutions come in to help maintain pH by consuming or releasing H to maintain that homeostasis, to maintain that balance. All right, take a moment to read this. If you need to, pause the video, pull up the PowerPoint, look at the, or rewind the video back to the pH scale. So which is the most acidic solution? The most acidic solution is one with what? And the answer is an H plus concentration of 10 to the negative 2. Now, don't let this fool you because 14 is bigger than 2. 10 to the negative 2 means that we're moving the decimal 2 places here, so it's 0.10. Whereas 10 to the negative 14, we would have to move 14 places, so it would be a very, very, very small number. So don't let that 14 and 2 throw you off. And a pH of 12 is actually alkaline. pH of 3 is an acid, but it's 10 to the negative 3. 10 to the negative 2 would be pH of 2. All right, let's go. All right, organic molecules contain carbon and hydrogen. We already talked about methane being one of our molecules earlier. This is a simple organic compound. It contains both carbon and hydrogen. I mean, it's a great example. We have 4 categories of organic compounds. We have carbohydrates, proteins, nucleic acids, and lipids. Lipids are kind of a special version. I'll get to that. Nucleic acids, we're going to talk about. We're going to get in more detail with a lot of these, but we're going to get even more detail with nucleic acids later because that's our DNA and our RNA. So we're going to talk about the basics of them now, but we're going to get more detail with them later. All right, so carbohydrates, proteins, and fats are common in our diets. Well, nucleic acids too, because you're eating other living things, so you're going to get DNA. We'll get to that. So mono means one, is a single unit of a carbohydrate. So we might talk about a specific, a monomer for a carbohydrate might be glucose. When we put the monomers together, we get polymers, poly meaning many. So you have individual pieces, and when you put those pieces together, you get bigger molecules. So dehydration synthesis, think about what dehydration means. If you are dehydrated, you don't have enough water in your body. Dehydration is taking away water. Synthesis is to make. So you're putting together 2 monomers by releasing a water molecule. So you have a monomer here and you have a monomer here. And you want to put these together to make a polymer, we need to remove water. So if we move this OH and this H, when we put those together, we get H2O, and we're able to connect these 2 monomers. If we wanted to put another one here, it'd be the same thing. We would have an H here, and then our, monomer, I'm not even going to try to spell that out, but these would have to join and be removed so that these 2 could link together. Hydrolysis breaks down to hydro and lysis. Hydro is water. Lysis is to break. Let's see if I can write it kind of quickly. Ooh, my handwriting is. You know what? Hopefully you can read it. So when we're doing this, we're breaking the polymers into monomers. So this is the opposite of dehydration synthesis. We are taking this water and we're going to break the water to break apart the monomer. So water gets broken into the OH and the H, to break this into two separate monomers. And this is what digestion is, breaking it apart so that you can put these into simple parts so your body can reuse those parts to make the things it needs. All right, so let's talk about carbohydrates. Carbohydrates, simple sugars. Monosaccharide is simple sugar, saccharide is sugar. Mono means one. Monosaccharides are the monomers of carbohydrates. So we have some here, ribose, glucose, fructose. Glucose is one you definitely want to be familiar with. Carbohydrates are these ring structures. Here they have carbon, hydrogen, and oxygen in them. So dehydration and synthesis binds two monosaccharides to make a disaccharide. So di means two. So when we combine glucose and fructose, so we have 1, 2 monosaccharides. We remove the water. So we remove the water to make sucrose. Sucrose you would know better as table sugar. So that's how we make it. And if we had, if you ate sugar, to break it apart, hydrolysis to turn it into fructose and glucose. Break it into simpler parts so your body can use it. Polysaccharides, poly means many, saccharide is sugar, are these long chains of carbohydrates. So first example is cellulose. Cellulose is the structure for the cell walls in plant cells. Starch is energy storage, like the starch in a potato. And you'll look at the starch granules in lab. And then glycogen is energy, so that's how we store energy in our livers. Stay there, when your body sends the right signal, some of it can be broken down and can help to raise your blood sugar. Back to that homeostasis at set point level. All right, so proteins are so diverse. They can be structural, such as collagen, like in skin, hair, and nails, or contractile, like the muscle proteins, myosin and actin. We can have transport, the membrane proteins found inside, in our cell membranes. We can have storage, like the egg protein. They can be digestive. Enzymes help to break things apart or put them together. And you also have some immune function. They're going to be tagged for like antibodies to help your body recognize things that shouldn't be in your body. Amino acids are the monomers of proteins. All amino acids have the same general structure. So you can see the structure here. I'm not too worried about knowing like the base structure, but definitely know that monomer of a protein is amino acid. We're going to get more into these when we get to protein synthesis. So I don't want to get super detailed into it now. So the R group is what really changes. The other basic parts are the same. You might recognize tryptophan, even though it's cut off there. Gets a bad rap for making you sleepy. It doesn't. Again, dehydration synthesis binds to amino acids, pulling that water out to form a dipeptide. Di meaning two. Peptide actually refers to the bond that's holding the amino acids together. So it's another specific type of bond. And then again, the function of the protein depends on its shape. We can have, this is what it looks like normally, and denatured means it doesn't work anymore. So cool thing about proteins is they kind of look like, ribbons. And then your denatured one kind of looks like a ribbon that you just kind of messed up when you're trying to, curl it with the scissors. And the example here is the egg just cracked out of the shell versus a cooked egg. Once you cook it, you're denaturing, you're changing the structure of those proteins. And they wouldn't work in the same way they would before. So we have primary structure where it's just a chain, and then secondary structure where it could start to fold into other shapes, whether it's the helix or the sheets here. Tertiary structure, this is the overall shape of one peptide. And quaternary structure, you have interaction between multiple polypeptides that make an even bigger functional protein. All right, nucleic acid, DNA, the Na stands for nucleic acid, and RNA, nucleic acid as well. So these contain genetic information. This is to help make proteins. So the monomers of nucleic acids are nucleotides. A nucleotide has three parts. It has a phosphate, a sugar, and a nitrogenous base. This sugar is deoxyribose. So this is DNA. If it was RNA, it would be ribose. That's one way to tell them apart between which nucleotide you're looking at. There are 5 types of nucleotides. You have adenine, cytosine, guanine, thymine, and uracil. DNA only has thymine is only in DNA, and uracil is only in RNA. We're going to get more into this when we get into DNA, and we talk about DNA replication, making RNA to make proteins. But definitely know these are the nitrogen bases that go with your DNA. These are in both. Thymine is in DNA only, and uracil is in RNA only. And again, dehydration synthesis links them together. Hydrolysis separates them. All right, lipids. Lipids are a little funny. So they are hydrophobic. Hydrowaterphobic, meaning scared of, they will not mix with water. They are energy storage, but they can also be waterproofing. They can be insulation. So now is the time of year when they start get ready to start showing them the bears fattening themselves up to go into hibernation, and they're storing their food as fat, but that's also going to give them the ability to stay warmer. It's going to help to insulate their body as well. So the example we have here, we have testosterone is an example of a lipid. So some hormones are lipids. We're going to talk about our phospholipids of our cell membrane. These are not built from chains of monomers, which is why you might not see them always included in the organic compounds, but they are important biomolecules. They form the basis of our cell membranes. Again, for us with hormones, they help with hormones for signaling between different parts of our body. So in order for a lipid to be made, you need a fatty acid and a glycerol. So there's actually 2 monomers, fatty acids and glycerol. So dehydration synthesis puts them together to make a triglyceride, and then hydrolysis separates them, the fatty acid from the glycerol. So fatty acids and glycerol are your monomers for lipids. They have the 2. You need both. Carbons of a saturated fatty acid are bonded to 4 other atoms. This is the difference between a saturated fat and a unsaturated fat. Unsaturated fat has at least 1 double bond, so at least 2 carbons are only bonded to 3 other atoms. It's not saturated. And the double bonds actually make kinks, and you can't really see this one kind of goes this way and then it kind of kicks out instead of being straight like these are straight. It prevents them from packing close together. So unsaturated fats like oils, this is what makes them liquids at room temperature. Trans fats have double bonds like the unsaturated fats but remain straight, therefore they are solid at room temperature. All right, sterols are important lipid molecules. Cholesterol is one that we hear of. It kind of gets a bad rap, but it's needed if you have the right one to help with our cell membranes. It helps to keep our phospholipids from clumping up too much or spreading out too much. And there's also some hormones that are just derived from it, like testosterone. All right, which monomer is incorrectly paired? Take a moment, pause, think about it, make a guess, see if you're right. Did you pick protein monopeptide? That's not a thing. Remember, protein is amino acid. Carbohydrates are monosaccharides, nucleic acid is nucleotide. Lipid, no real monomer, because there's not a single one. Remember, you need fatty acids and glycerol. All right, last little section. Aphids. Hopefully you really don't know much about aphids because they are a pain if you have a garden. So there's certain organic chemicals that account for color variation in these aphids, and you can see the days after birth how the color changes from 0 to 15 days after birth. Of course, somebody looked at this and said, what causes this? Let's take a deeper look into it. So they discovered that there's actually a bacterium, Rickettsia, I think I said that right, that causes this color change. So depending on if they're infected or uninfected, will help to determine their color change there. So when the aphid produces more green pigments, aphids eat plants. They latch onto them and actually suck nutrients out, which is why they're bad for your plants. But if they're green, then they can blend in because plants tend to be green. So of course, if they're red, they stick out more. So that means the predators are going to be able to easily spot them because they're going to contrast with the green plants that they're on, which is good if you want the predators to eat them. I actually, little side note before I end this video, I have milkweed plants, and there's a particular aphid that goes on them, and they are bright yellow. Well, milkweed plants are green, so you can see these aphids very distinctly. And I would, wash them off. I wasn't doing too much. I did have some bugs. I didn't figure out what they were yet, but the bugs have taken care of them because they're gone. About a month and a half ago, they were pretty badly infested, but they were so visible. I was actually surprised that nothing ate them, but something came in and ate them all, and I haven't seen a single one in a couple weeks. So natural pest control. But going back to this, if the infected one is causing it to produce more green pigment, then it's allowing it to blend in better. So the infection actually gives it an advantage. It allows it to produce more green pigment, allows it to be more camouflaged, less likely to be eaten by predators. All right, so that is chapter 2. Hopefully that helped you out, especially since we didn't go through all of this in class the other day. If you have any questions, be sure to email me.